Hello, everybody. I hope you enjoyed your lunch. I did very much. And I would like to invite you for our afternoon session. And I would like to invite Violaine, who actually works right around the corner in the Natural History Museum in Paris. And she's working on the morpho butterflies, but she studied her PhD on Arabidopsis halleri, which is very nice. And then she also continued her postdoc in Montpellier and in the UK. And in general, she's interested in the role of balancing selection and adaptation. So the floor is yours.
Thank you. Hello, everybody. I'm actually very happy to be here for this last-minute talk. So thank you, Abdou and Virginie, for thinking about me. That's very nice. So today I'm going to show you some results we've got on the diversification of morpho butterflies in sympatry. So I would be more in the ecology side of the spectrum. I hope you don't mind. So morpho butterflies are found in the Amazonian rainforest, which is, and this is something you probably all know, a very species-rich environment, and which is a very nice playground for us evolutionary biologists to question the kind of feedback you can have between ecological interactions on one hand and the evolution of traits and speciation. So whether the evolution of new species changes ecological interaction you can have among those species in sympatry, or the other way around, how much ecological interactions are shaping the evolution of traits and the diversification of species. So we are focusing on those very nice butterflies of the genus Morpho. They can have different wing shape, wing colors and sizes, and also different ecologies. So some species are found mostly flying up high in the canopy, while others are found mostly in the understory microhabitats. And we are interested in understanding why they coexist and what are the evolutionary processes leading to those different species with different phenotypes. So in the morpho species living in the understory, we frequently have this kind of color pattern, so very contrasted color pattern with iridescent blue dorsal patterns and brownish cryptic coloration in the ventral side. So just for your own information, so this iridescent coloration stems from the specific structure of the wing scale that are found on the dorsal side. And the consequence of this very specific dorsal coloration is that when they display their typical flap gliding behavior, we have those flashes of blue. So here it's in slow motion and here in the actual speed. And basically, this has a very confusing effect on predators and also on the clumsy butterfly hunters like me. And so this combination of a fast erratic flight and a specific wing color pattern enhance the confusion of predators and enhance the escape abilities of those butterflies. And so we know that birds can learn to associate the blue coloration and these escape abilities. And so we think that they can learn and they can then focus on alternative prey species. So the blue coloration can become a signal of evasive capacities. And this will generate what we call positive frequency-dependent selection on color pattern, similarly as in Müllerian mimicry, even if those butterflies are non-toxic. So we wanted to test whether the evolution of color patterns depends on the density of different morpho butterflies living in sympatry. So especially, we focus on 3 closely related species of morpho, so this morpho Achilles, morpho Eleanor, and morpho Daidamia. And we use the museum collection, so we are lucky enough in the museum to have more than 7,000 morphospecimen in our collection. So we were able to sample the whole South America while staying in Paris, basically, with this museum collection. And we look at the variation in the width of the blue band on the dorsal side of the wings in these 3 species. So here is the spatial distribution of those 3 species. So Deidamia and Achilles are basically restricted to the Amazonian basin, while Morpho-Eleanor has a larger distribution going until the south of Brazil and up to Panama and Central America. And what we detected here using those museum specimens is that there is a striking convergence, local convergence in the width of the blue band throughout the 3 species. So basically, in French Guiana, they have a very thin band, while in Peru, for instance, they have a much larger band in all 3 species. And we think that this parallel evolution of wing color pattern in those 3 species is driven by the predator behavior and the escape mimicry phenomenon. So we have this, we described in this paper the mimicry between those species living in sympatry, probably due to predator behavior. And then, because wing color patterns are widely used as mate recognition in diurnal butterflies, we question whether this similarity in color pattern results in reproductive interference between those different species living in sympatry. So to test that, we set up an experiment in Peru, so in the Amazonian part of Peru, and this was the work of a PhD student named Camille Leroy. So what Camille did, he made a very fancy experiment where he took actual wings of Morpho and he glued it on this solar-powered system. It's something you can buy in every for your garden. So basically, there is a solar panel, and it makes the butterfly move. So he put this device on the bank of a river. So Morpho males usually patrol along those rivers, and we have 4 sticks, 1 meter high, which were used to define what we call an interaction space. And we had 2 cameras to film the butterflies. So basically, this is another point of view. So here is the device, here is the river, and you can have wild males going up and down on the river. So basically what we record is whether the males were just passing, whether they were changes of trajectory to approach our dummy butterflies, or whether they enter into our interaction space and interact with our dummy butterfly. So we had different dummy butterflies from male and female of the 2 sister species Morpho Achilles and Morpho Eleanor, and the wings were rinsed with hexane to remove every potential pheromone interactions. So we were just testing the effect of the visual signal. And basically, so here are the main results. So here is the male, so conspecific and heterospecific dummy butterflies. And here when the visitor was Morpho Eleanor male or visitor Morpho Achilles males. And here you have the percentage of interactions with male dummy. So basically, they come very often to interact with our dummy butterflies. And we had no effect on the species of the dummy butterfly. So they interact with male from both species. So when the dummy butterflies were females, here Morpho Eleanor males, they interact with both their conspecific and heterospecific females. But for the male Achilles, they were a bit more discriminant. They were preferentially interacting with their conspecific. But altogether, this experiment documented a very high reproductive interference between those sympatric species, probably enhanced by their color pattern similarity. So then what we wanted to test is to test for the level of encountering between those species, because it will modulate the level of reproductive interference. So what we did again with Camille was a very simple capture, mark, recapture experiment. So basically you capture the morpho when you succeed. You put a number on the wings and you just note the species, ID, time, and day. And then you do that for several weeks, and you look at the recapture rates. So this allowed us to discover something very interesting, is that so for those two sister species, Morpho Eleanor in light blue and Morpho Achilles in dark blue, we had like similar densities. It's again in the same river in Peru. But the timing where we capture the male from the two species was different. So we had Eleanor earlier in the afternoon as compared to Morpho Achilles. So potentially this temporal segregation limits the reproductive interference between those two species. So you have a kind of a story that you can build up with this. So because you have mimicry, color pattern mimicry in those species living in sympatry, this can enhance reproductive interference, but this can also favor the evolution of different temporal niches. So this is a question we are currently working on. How much the fact that you have two different species and that the hybrids are probably not viable, does it exert some selection on the temporal niches of those sympatric species, or whether the evolution of different temporal niches fuel speciation in sympatry? And of course, to explore this question, we need to look at the impact of potential genetic incompatibilities that can arrive through time in divergent population. So this is a work we are currently doing with Titouan, who's a master student, who's in the room. So basically, what Titouan and a previous student have been doing is to build an individually centered models and to run stochastic simulations. So we have, so here are the main assumptions. So we assume two species, and we assume that hybrids are completely non-viable, non-fertile. So there is a high cost in females when they mate with heterospecific male. And we assume mimicry between our two species that trigger important reproductive interference because of male-male competition, male-male interactions. So we have a kind of density-dependent selection acting on the patrolling behavior. So basically what we assume is that we have different timing of emergence of our butterflies between species 1 here and species 2. And then we are looking at the evolution of the patrolling window of male, which is centered around a value we call HA. So we look at the evolution of the position of the window and also of its breasts. So if we assume that there is a single species and we have an emergence, let's say, in the morning, if we look at the evolution of the patrolling window of males, what we will observe is that most males will patrol close to the emergence peak of females, because in those butterflies we assume that female can mate only once, while male can mate multiple times. So that virgin females are really the limiting factor for those males to reproduce. So basically males will patrol just after the female emergence with a mean timing of patrolling just after the emergence time. So this is when you have a single species, and so maybe I will skip that. But then when we assume that there is two species living in sympatry, first what we looked at is the probability that both species coexist, depending on the temporal distances between the emergence peaks of the two species. So basically, when female of the two species are emerging at similar timing, you barely observe maintenance of both species in sympatry. As soon as you start to assume a larger distance between the different timing of emergence of the females, then you can have coexistence between species in sympatry. So then what we look at is the distance of the male patrolling window from the emergent peaks of the female. And we plot that, and here it's a distance between the emergence peak between species. So as you can see, there is a large effect of the timing of emergence of the virgin females on the difference in temporal window between species. So maybe I can explain with a scheme. So basically what we add in allopatry, when you have a single species, you have here the peak of emergence of the female, and here the peak of patrolling activity in dotted line here for species 1 and for species 2. So this is like in allopatry. And then if you consider sympatry and if you allow for reproductive interference, what we see here in this area, so when the emergence peaks are not exactly the same but still quite close in time, is an evolution of the male patrolling window. So in the earlier species, males tend to patrol earlier than the female emergence peaks, and this is the other way around in the species 2. So basically, there is a huge effect on the reproductive interference in the temporal niches of those two species. And we also had an effect on the width of the patrolling window. So again, we had a smaller width in allopatry that tends to be larger, as you can see here, when the two species are in sympatry, also because the patrolling the position of the patrolling window is slightly earlier, for instance here, from the emergence peaks of the female. So basically what this model and those simulations are showing is that there is a strong effect of sympatry on species coexistence, but also on the temporal niches. So we can draw a kind of causality link between speciation and reproductive interference and the evolution of temporal niches in those sympatric species. But then we may ask, but can it be the other way around? Can speciation be fueled by the evolution of different divergent temporal niches? So what we did with Titouan is to extend the previous model. So we made two main changes. So now we are not considering that only males are patrolling. We are considering that there is a window of reproductive activities restricted during the day in both males and females, and that will influence their probability of mating, encountering and mating. And we assume B loci, and there are there is two possible alleles, 0 and 1. Those alleles are completely neutral in terms of there is not one allele more adapted to the earlier timing than the later timing, but they can be genetic incompatibility when you are heterozygote as those loci. So this kind of neutral Dobzhansky-Müller incompatibilities. Sorry. So here are the results of our simulation. Without genetic incompatibilities, we are looking at the evolution of the temporal window of males and females after 500 generations. So here is the timing of the day, and here is the density of individuals having different patrolling periods. So here we assume that the emergence of both males and females is in the middle of the day. So you can see that the patrolling or the timing of reproductive activity is still centered around the emergence times of those butterflies. But we can see that there is a kind of double bimodal distribution. And we think it's driven by the male-male competition we assume in the model that generates negative frequency-dependent selection on the position of the temporal window. But still here, it's slightly bimodal, but we do not have a too well-separated population of our morphos. Then if we add some genetic incompatibility, so again, assuming that offspring become inviable when they are heterozygote, here, for instance, at 1 out of the 10 loci, but it doesn't change much how much the threshold you put for incompatibilities. Then what we observed after 500 generations of our simulation is that we can have an actual bimodal distribution in the timing of reproductive activities in our initial population. So we observed this emergence of two subpopulations. This is exactly the same parameter used in both simulations. The only thing that changed is that we assume those incompatibilities. So what we can do now is compute, for instance, the FST between our populations, and depending on the intensity of male-male competition. So here you have I think a few 100 simulation done, and here you have the mean value of FST. So you see that without incompatibility, we still have very low FST values, but still it does increase when you increase the intensity of male-male competition, because again this tends to create this bimodal distribution in the timing of reproductive activities. But if you add incompatibilities, then you can have a much higher FST obtained. So just to conclude, so my hope was to convince you of the importance of eco-evolutionary feedback that we can observe in sympatry. So here, typically, we can see that ecological interaction in sympatry, so the relationships with predators, but also the heterospecific male-male aggression, are driving the genetic and phenotypic evolution and may fuel speciation. So in that direction. But in the other direction, when we assume some genetic incompatibilities, this can also shape ecological interaction, modify the ecological niches. So this was just to illustrate how, with our specific case study of morphobutterflies, we can observe those interesting feedbacks. So I just want to thank you for your attention and thank the whole Morpho team. And just a short advertisement. So we are looking for a postdoc to continue on this Morpho project. So if you know anyone interested, please give them the note. Thank you.